I'm Priya and working as an assistant professor in Biochemistry from St.Joseph College of Arts and Science, Cuddalore. I'm going to share on the topic nitrogen fixation and NIF genes in Klebsiella pneumonia. To start with, biological nitrogen fixation. The reduction of atmospheric nitrogen into ammonia by soil-borne microorganism is called biological nitrogen fixation. It was discovered in legumes by Wilfred. The global nitrogen fixation is 175 into 10 to the power 6 tons per year. Nitrogen is one of the major elements useful for plant growth. Our atmosphere contains about 80% gaseous nitrogen, but green plants are unable to use it directly from the atmosphere. Some soil bacteria and blue-green algae are capable of reducing the atmospheric nitrogen into ammonia in their cells. The process of nitrogen reduction is called diazotrophy or nitrogen fixation. Diazotrophs are broadly classified into two groups. One is non-symbiotic nitrogen fixers, example Azotobacter, Clostridium. Symbiotic nitrogen fixers, Anabaena, Noastac, Rhizobium, etc. Non-symbiotic nitrogen fixers. Some microorganisms live independently in the soil and do nitrogen fixation. They are of two groups. They are free-living autotrophic diazotrophs and free-living heterotrophic diazotrophs. In free-living autotrophic diazotrophs, they synthesize their own food by photosynthesis from sunlight and or by other chemicals. Among these, some are aerobics, example Oscillatoria, Cylindrospermum, Plectonema, etc. And anaerobics include Chlorobium, Vibriform, Rhodosodomonas, Viridis, etc. And free-living heterotrophic diazotrophs include dead organic matter as food and do nitrogen fixation. Example Clostridium, Azotobacter, Klebsiella pneumonia, etc. So features favorable for non-symbiotic nitrogen fixation. All diazotrophs contain the enzyme nitrogenase which catalyzes the reduction of nitrogen into ammonia. And this nitrogen is sensitive to oxygen, so it prefers anaerobic conditions for nitrogen fixation. But in microbes, the oxygen level is usually high. And the high oxygen level leads to oxidation of nitrogenase and hence that enzyme becomes inactive. So non-symbiotic nitrogen fixers have the following special features for nitrogen fixation. So this is the first special features for nitrogen fixation, that is spatial separation of nitrogen fixing cells and photosynthetic cells. Here blue-green algae are photoautotrophic diazotrophs living on moist soils. They have photosystem 1 and 2, so that oxygen is evolved during the light reaction of photosynthesis. This oxygen may inhibit the nitrogenase activity. In these algae, photosynthesis is restricted to vegetative cells and nitrogen fixation is restricted to heterocyst. Example Anabaena, Noastac, Cylindrospermum, etc. So here heterocysts are thick-walled cells that lack photosystem 2. The thick wall of the heterocyst prevents the entry of oxygen into heterocyst. Since photosystem 2 is absent here and the oxygen has not been evolved during photosynthesis in the heterocyst, but respiration is going on continuously. Because of these reasons, partial anaerobic conditions develop in the heterocyst for nitrogen fixation. Heterocysts have various enzymes involving in the anabolism of urides. So next one will be protein nitrogenase association. They are aerobic heterotrophic nitrogen fixing bacteria. In these bacteria, some intracellular proteins are found associated with nitrogenase when oxygen level is high in these cells. When the oxygen level comes down, the proteins release the nitrogenase free, and the free nitrogenase fixes nitrogen as usual. So the next one will be high rate of respiration. Some aerobic heterotrophic diazotrophs show high rate of respiration. So the cellular oxygen level is reduced to a certain extent. At the reduced oxygen level, nitrogenase reduces nitrogen into ammonia. Example will be Azotobacter. And the fourth one will be time-specific nitrogenase activity. It is an aerobic photoautotrophic diazotroph. It does photosynthesis in the daytime and does nitrogen fixation in the night. In the night, oxygen level comes down in the cells due to continuous respiration. And last one will be association with rapid oxygen consumers. Some microbes cannot fix nitrogen when they are living far away from rhizosphere zone of some higher plants. Example, Klebsiella, Rhodosudomonas, Chlorosudomonas, etc. When they are living in rhizosphere of high oxygen consuming plants, they fix the atmospheric nitrogen. Here, plant roots create a partial aerobic or anaerobic conditions that favors for nitrogen fixation. Mostly, anaerobic condition is preferred in this association with rapid oxygen consumers. So this slide shows about the symbiotic nitrogen fixation. Some microorganisms establish symbiotic association with plant roots and do nitrogen fixation. And during this symbiotic association, some diazotrophs induce nodal development on the plant roots. This type of symbiosis is called rhizocenosis. Example, rhizobium. Some symbiotic diazotrophs do not form root nodules in host plants. For example, Azospirillum, Azotobacter, etc. So symbiotic diazotrophs cannot fix the atmospheric nitrogen when they are living alone in the soil, but certain cyanobacteria are fixing nitrogen both when they are residing in the host and when they are alive free in the soil. Example, Nostoc, Anabena, etc. Host specificity. Although several species of Rhizobia live in the rhizosphere of a legume, a particular species alone can establish symbiotic association with its roots. This selective infection of Rhizobium on specific plants is called host specificity. And the host specific infection of Rhizobium depends upon the specific flavonoid secreted by the roots of legumes. Alfalfa exudes luteolin and white clover exudes dihydroxyflavin along the lectins. Here, alfalfa is a herbs which is rich in calcium, phosphorus, etc. and it reduces the cholesterol in the intestines. The root exudates induce certain genes of particular species of Rhizobium to produce a host determinant compound on its cell wall. This slide shows about the mechanism of host-specific infections of Rhizobium trifoli on the root hair of alfalfa. In most cases, the host determinant compound is a capsular polysaccharide. In short, it is CP. The lectin produced by the legume root has an affinity to the capsular polysaccharide. Therefore, it binds with the capsular polysaccharide of the specific Rhizobium species and the other end of the lectin binds with the cell wall polysaccharides of a root hair. Here, lectin acts as a bridge molecule. After recognition, this Rhizobium infects the root hair. This slide shows about the genetics of nitrogen fixation, and NIF genes in Klebsiella pneumonia is an example for this. All organisms including humans, plants and other organisms demand nitrogen for their daily cellular activities. Many cellular components are also made up of nitrogen. Nitrogen fixation is the essential biological process and the initial stage of the nitrogen cycle. So nitrogen fixation refers to nitrogen in the atmosphere which is converted into ammonia by certain bacterial species like Rhizobium, Azotobacter and by other natural phenomena. So what is NIF genes? Nitrogen fixation in diazotrophs is genetically controlled by a cluster of genes called nitrogen fixing genes or NIF genes, and they code for the enzyme nitrogenase that reduces nitrogen into ammonia. The nitrogenase enzyme plays a role in the useful of nitrogen fixation and it occurs in chromosomal DNA, but in most symbiotic diazotrophs they are seen in extrachromosomal DNA called megaplasmid and they vary from species to species. This slide shows about the NIF genes of Klebsiella pneumonia continuations and it is a free living diazotrophic bacterium. The NIF gene cluster is 24 into 10 to the power 3 base pairs in length and 18 into 10 to the power 6 Daltons, that is molecular weight. It lies between the genes coding for histidine and uptake of shikimic acid and it consists of 17 genes. They are listed here. This slide shows about the structure of NIF genes in Klebsiella pneumonia. The expression of the 17 NIF genes in Klebsiella is regulated in response to the nitrogen source by the products of the nitrogen control genes. And the NIF transcription is also specially regulated in response to oxygen and low levels of fixed nitrogen by the products of NIF-LA operon. This operon is made up of linked genes, which is thought to regulate other genes responsible for protein synthesis. So this slide shows about the regulation of NIF genes. NIF K and D encode for nitrogenase enzyme. Q participate in molybdenum uptake during the biosynthesis of nitrogenase enzyme. NIF B, N, E and B involved in the biosynthesis of ferrous molybdenum. S involves in maturation of nitrogenase by combining the products of NIF, K, D, Q, B, E and B. So here NIF-H encodes for nitrogenase reductase, that is iron protein. M involves in the modification of primary transcript of NIF-H. F codes for a flavor protein. J codes for pyruvate flavodoxin oxidoreductase enzyme. A activates the transcription of NIF genes cluster. L codes for a protein that suppresses transcription of NIF genes when oxygen or ammonia level is high. NIF U, X and Y is found in gene map. NIF genes control the expression of NIF genes when there is scarcity for ammonium and glutamate. That is low amount of ammonium and glutamate, product of NTRC, that is nitrogen regulated C gene, bind with NIF-A and activates the NIF gene cluster. And next point will be the concentration is high in the cell. It is said to be repressing the transcription. That is, if their concentration is high in the cell, the product of NTRB gene binds with the product of NTRC or NIF-A and repress the transcription. Likewise, the product of NIF-L, when oxygen or ammonium level is high, it binds with the product of NIF-A and stops transcription. And according to Zhu and Brill, it was suggested that the temperature above 37 degrees Celsius represses the transcription of NIF genes. Thank you.